Hi everyone. Now we are going to discuss about influenza or the flu, which is one of the airborne diseases. Influenza or the flu is an acute contagious disease of the upper respiratory tract transmitted by airborne droplets. And the disease is believed to take its name from the Italian word called influence, a reference either to the influence of heavenly bodies or to the influenza di freddo, which means influence of the cold. At one time, it was thought to cause it by a bacterium called Haemophilus influenza. Later on, it was discovered that it's not the bacteria, it's all because of the virus, and that's why it was named as influenza. And this is going to occur either as a sporadic or epidemic and the pandemic form. Influenza virus belongs to the family Orthomyxoviridae. The name of this orthomyxovirus refers to the affinity of the virus to mucins. That is, mixa means mucus, which is able to adsorb on the mucoprotein receptors of RBC, causing hemagglutination. And these influenza virus were also going to be classified under single stranded RNA virus as their genetic material is RNA. Now coming to the causative agent, the influenza virus, as I told you, belongs to the Orthomyxoviridae family, and these are going to be of a spherical in shape with a diameter of 80 to 120 nanometers. And this is going to have internally 8 single stranded segments of RNA of different lengths. The nucleocapsid is surrounded by an envelope. So where is your envelope? This gray color one is going to be of envelope, which consists of an inner protein layer. So this pink color is a protein layer. Okay, and outer is going to be the lipid layer. So inner layer, protein layer, is going to be coded by the virus, and the outer lipid layer is going to be coded by the host cell membrane. That is, it is derived from our cell. And if you observe here, there are two types of the spikes. One is going to be this one and one is this one. So one spike is going to be called as heme agglutinin spikes, and the other one, this mushroom shaped one, is going to be called as N spikes or neuraminidase spikes. Okay, and these two spikes are very important in their role. For example, take this heme agglutinin spikes cause agglutination of RBCs. An important feature of the heme agglutinin of influenza virus is that antibody directed against it prevents the virus from infecting a cell. Thus, antibody directed against the hemagglutinin, that is against this one, neutralizes the virus, and this is the mechanism by which that is our immunity to influenza is brought about during immunization process. Then coming to neuraminidase, that is N-spike, is an enzyme which breaks down the sialic acid component of the cytoplasmic membrane. On the inner side of the envelope, there is a protein called, here you can see the M1 protein and M2 protein, which provides rigidness and thus stabilizes the lipid bilayer. So this is all about the hemagglutinin spike and the neuraminidase spike. So the neuraminidase spike is going to be of mushroom shaped one, and here is going to be the pointed one is the hemagglutinin. Okay, then as I told you, we are going to have three types of influenza depending upon their antigens H spike, that is hemagglutinin spike, and the neuraminidase. These influenzas are being classified into three types: influenza A, influenza B, and influenza C. Out of the three groups, the influenza A is the most important human pathogen. Okay, so let's move to the replication. So the replication or the more number of viruses is going to be enhanced by the process called as endocytosis. That means influenza virion enters into the host cell by the process called endocytosis. And there in the cytoplasm, the nucleocapsid separates from the envelope and moves into the nucleus, where here, that is our nucleus, where in the viral genome replication takes place. Uncoating results in the activation of the virus RNA polymerase. So this virus contains an RNA polymerase enzyme, okay, which is going to be activated once the virus is uncoated. Now the mRNA molecule, so here through the process of transcription, the mRNA molecules are then transcribed from the RNA virus, okay, inside the proteins are synthesized in the cytoplasm. Nearly 10 virus proteins are encoded by the 8 segments of viral genome. Some of these proteins are involved in the viral genome, uh, what we call it as viral genome replication, while others are involved in the structural components or structural proteins. And these are all going to be used in the formation of the virion particles. Okay, and now what happens? The formation of the complete enveloped progeny virion takes place by the budding process. So this is all about the replication of the influenza virus. So they will enter into the cell by the process called as endocytosis, and from there uncoating of this virion part, that is envelope, is going to occur, and the nucleic acid will enter into the nucleus of the cell, and there the RNA replication occurs and synthesizes the mRNA. Now this mRNA moves into the cytoplasm, and where there it takes place the synthesis of all the protein machinery that is essential for the formation of the structure, and after that they're all being released into the cell through a process called as budding process. So this is all about the replication of influenza virus. Then moving to the epidemiology. So here epidemiology is going to be of mainly it is going to be of either, I told you already, that is sporadically, or it may be epidemic or in the pandemic forms, isn't it? And the source of infection is an infected individual, and the virus is shed in the respiratory secretions short, that is before the onset of illness, and for about 3 to 4 days thereafter. And the subclinical infections are common. Influenza virus type C is endemic throughout the world and causes very mild or inapparent infections. Whereas coming to the influenza type B is going to be of causing sporadic as well as epidemic influenza. And while coming to the type A, as I told you, most pathogenic to humans, can cause pandemic as well as sporadic as well as the epidemic. That means it is able to cause the sporadic, epidemic, and pandemic. In 19, that is the most severe pandemic in the human history is going to occur between the 1918 to 1990, during which 20 million people were perished. And then an unusual picture, that is of a feature of this pandemic, was the very high rate of mortality among the young adults rather than all of the age. Young adults were mostly affected. The next pandemic was occurred in 1957 when the Asian strain originated in China and spread throughout the world within a short period. In 1977, epidemic influenza appeared in China and then in Russia, hence called the name as red flu. Okay, and this is all about the epidemiology. Then moving to a little terms which are very important, that is antigenic drift and antigenic shift. The reason why the virus able to cause epidemics and pandemics is its ability to grow, that is to undergo what we call it as antigenic variations. That means often it is going to change its antigenic nature. So those are being called as antigenic variations. So there comes the antigenic drift and antigenic shift. Coming to the antigenic drift. The antigenic drift or minor annual variations. So very minor variations. So here you can see minor variations resulting from mutation and selection and is responsible for epidemics. So here you can see the spike is going to have very little change. The rest of the thing is same. So if you are having that minor annual variations resulting from mutation and selection, and that is responsible for epidemics especially. Then coming to the antigenic shift. The antigenic shift is going to be occurred by the genetic recombination. So here it's a completely different. This is how you are going to have your hemagglutinin. But here you can see it's completely different, resulting in a new virus strain due to antigenic alteration of the spikes and is responsible for pandemics. The drift is responsible for epidemics and the shift is responsible for pandemics. Okay, so this is all about the things that you have to remember. What is a drift and what is a shift? Drift is because of the mutations and shift is because of the recombinations, and here because of the recombination which is resulting in completely a new virus strain and responsible for causing the pandemics. Then coming to the pathogenesis, obviously, which is very important. The virus is going to be acquired by the inhalation of the virus contaminated respiratory secretions. That means when anybody is going to sneeze, the infected person is going to sneeze, cough. So obviously those droplets contains this virus. So if you are going to inhale that one, so obviously you are getting infection. The viral neuraminidase facilitates infection by reducing the viscosity of the mucus film lining the respiratory tract and exposing the cell surface receptors for virus adsorption. So we have discussed that there are two types of the spikes. One is going to be the neuraminidase spikes and another one is the hemagglutinin spikes. So these are responsible, which is going to reduce the viscosity of our mucus film lining the respiratory tract and exposing the cell surface receptors for virus adsorption. And then what is happening after that? The ciliated epithelial cells of respiratory tract are the main sites of infection. And now these epithelial cells are damaged and shed, laying bare the basal cells in the trachea as well as on the bronchi. Okay, and this renders the respiratory tract highly vulnerable to secondary infections with bacteria such as Haemophilus influenza, pneumococci, and then streptococci, and then staphylococcus aureus. So these are all the things that are happening once this pathogen enters into our body. Then what are the symptoms that we are going to come across when we are having this infection? So the incubation period of this virus is going to be of about 1 to 3 days, and the disease varies in the severity from a mild coryza to a fatal pneumonia. The flu is characterized by having some sort of what we call as a fever, then chills, and then headache, malaise, and general muscular ache. So these are all the symptoms of a mild flu. Okay, and recovery occurs within the 3 to 7 days, during which cold-like symptoms appear as the fever subsides. So these are all the symptoms where we find when we are having this flu. Then coming to the laboratory diagnosis, that means how we are going to diagnose it. The diagnosis consists of demonstration of virus antigens and then isolation of the virus and serology. So these are the methods by which we are going to we are going to demonstrate that the virus is going to be present in so on so patient or not by these techniques. So demonstration of the virus antigen is done mainly by immunofluorescence technique, then RIA and ELISA. And even smears of the nasal swabs and nasopharyngeal secretions or centrifuge deposit of throat garglings are prepared on the slides and stained by immunofluorescence method and examined under fluorescent microscope. So if we are going to find the virus, then obviously we can give the report as they are having the influenza infection. Then the second type is the serology, where complement fixation test and hemagglutination inhibition (HI test) are conducted to demonstrate the antibodies to influenza virus in patients' sera. So these are the two main techniques by which we are going to diagnose the disease or infection. Then coming to the prevention and control. So obviously immunization is necessary for only when a pandemic is anticipated. isn't it? But subunit vaccines containing hemagglutinin and neuraminidase subunits have been introduced because the older method of taking the virus, that is the whole vaccines, was going to have some sort of a side effect. So recently we are having some sort of the preparations from subunit vaccines, which are a good one being used widely nowadays. Now, the immunoprophylaxis is difficult due to the frequent change in the antigenic makeup of the virus. Along with this immunoprophylaxis, we are also having the chemoprophylaxis, which means preventive chemotherapy. So, this is the other device used for the prevention. So, amantadine, rimantadine, the synthetic amines are given orally to the immunized, that is the person who is going to have a very less immune, that is unimmunized, high risk individuals during a major epidemics. These antiviral drugs block the viral M2 protein, which functions as an ion channel. Not only that, they act only with the influenza virus and not with the influenza B virus that lacks the M2 protein. So in the structure we had gone through the M1 and M2 proteins, isn't it? So here our drugs, that is chemotherapy drugs, are mainly influencing the M2 protein. So this influenza A virus is going to possess this M2 protein, but influenza B is not having this one. So these drugs may not be useful to the preventive treatment in the influenza B virus. And the treatment of influenza symptoms with aspirin, especially in children younger than 14 years, is not recommended because they are going to have some sort of diseases like Reye syndrome are going to occur, and even some young children are going to die because of usage of these drugs. For that reason, these drugs are not recommended for the children younger than 14 years. Okay, so this is all about the what we call it as prevention and control of influenza. Okay, thank you.